Good afternoon, everyone. It's my pleasure to introduce Siqi Liu, who is an assistant professor in the Department of Pharmacology. Dr. Liu is a homegrown faculty member. She did her graduate work in James Chen's lab, where you may remember this couple of famous papers. She described the mechanism by which the antiviral protein MAVS activates the innate immune system in response to a viral infection, and then went on to show how that activation is regulated by phosphorylation and the interaction of the proteins involved in that pathway. She then went on to an equally productive postdoc in Elaine Fuchs' lab at the Rockefeller University as a Jane Coffin Childs Fellow, which was a big honor. And there she showed an important link between wound healing and the immune system, some of which she's going to talk about today. And based on that work and her own ideas, Siqi received a K99 award. And last year, we were pleased to recruit Siqi as both an endowed scholar and a CPRIT scholar, which is one of the more difficult things to be able to do for a recruitment. In her own lab, she will continue to explore how epithelial tissues sense and respond to injury. Siqi, it's up to you to show us how.
All right. Thank you, David, for that kind introduction. Thank you, everyone, for coming here. I'm very excited to be here, and I want to share with you some insights I've gained over the years on understanding tissue injury repair, how we think of it as a host response to homeostatic disruption. The ability to maintain homeostasis is essential for life. However, we constantly experience disruption of such homeostatic conditions, which we propose there are conserved mechanism for sensing and respond to such disruption for us to return back to homeostasis. Pathogen infection and tissue injury are two universal stimuli that quickly disrupt homeostasis. When we know more about pathogen-derived non-self patterns such as bacterial DNA, viral RNA, activate host response that leads to cytokine production and ultra-strict host defense. We know much less about how we sense injury, which then generate a proper response for host repair. We use mammalian skin wound healing as an initial model, which we think is a good model to study injury repair, as you can think that skin as an organ that we constantly experience injury, and it has robust repair ability. It also is representative of most epithelial organs. Importantly, as you can show from here, that when the open wound occurs, it actually needs a localized response that coordinates both epithelia and dermal mesenchyme signal for a tightly coordination in time and scale for a proper repair. We have very versatile genetic tools to study this question, and more importantly, aberrant skin wound repair affects up to 20% population, and its associated pathology can lead to infectious disease, inflammatory disease, and cancer. Here, I'm just going briefly tell you the story that we stepwise found that so tissue injury sensing leads to a damage sensing pathway that leads to IL-24 production, which then activates a transcription factor, STAT3, that is essential for host repair. This started with first identify STAT3 as an essential factor for wound healing. Here is a sagittal section of a skin wound healing in the Y type. The green labeled skin epithelium re-epithelialize and close the wound in a time course of several days. When we delete STAT3 in the skin epithelium, it disrupts this re-epithelialization process, suggesting that STAT3 as a master transcription factor is essential for wound healing. Well, we know that pathogen-induced non-self patterns elicit this host response due to production of type I interferons, which then activate a master regulator STAT1 for host defense. I propose that the role of injury-induced STAT3 in host repair is reminiscent of pathogen-induced STAT1 in host defense. So first, we set to answer what upstream injury signal that induce STAT3 activation. We took a piece of mouse skin wounds and searched for STAT3 activating cytokines that induced in the wound. One cytokine here stands out, which is IL-24. So IL-24 is a cytokine that is not expressed in the homeostatic condition, but remarkably induced after injury in the skin epithelium. And we also show that IL-24 is specifically expressed at the wound edge epithelial stem cells. What happens when we don't have IL-24? We generated IL-24 non-mice. As you can compare wild type versus knockout in the healing wounds. So the green labeled epithelial stem cells trying to migrate into the wound bed to close. And accompanying this is a robust dermal proliferative response, including angiogenesis. However, when you don't have IL-24, all those epithelial cells is still able to re-epithelialize through a thinner layer. The dermal response accompanying it is completely abolished. This suggests that epithelial-derived IL-24 is essential for coordinating the epithelial and the dermal response for tissue-level repair. Now, what injury signal induce IL-24? We first exclude the possibility that the pathogen or commensal bacteria in the skin is essential by performing germ-free mice, and this pathway is still intact. Later, we hypothesize that it is an injury-induced pattern that normally don't present in a homeostatic condition that induces pathway. So we test a panel of possible injury patterns that normally present in the wound, and we found that among all these patterns we tested, tissue hypoxia is one pattern that is induced by the injury that can robustly induce IL-24. And here I want to show you that. So these are not only analogous pathways, but we propose these are homologous and common ancestral pathways to resolve disruption due to that both pathogen infection and injury induce patterns that normally don't exist in the homeostatic condition. And they both induce interferons versus IL-24. And interestingly, these cytokines families, they are the most closest evolutionary homologs right next to each other in a phylogenetic tree. Same as their receptors, same as these transcription factors. So this suggests that, so these two pathways, they actually share a common ancestor back in evolution in response to homeostatic disruption, and then they later diverged into separate pathways dealing with more specific response. So this paradigm will help us to further understand how injury response is actually an innate immune response. Going forward, my lab is interested in studying and understanding how epithelial tissues sense and respond to injury. Injury site is a fascinating place. When you have an injury, it disrupts the tissue architecture and also the nutrient supply. So it becomes resource limiting. But well as you want to repair the injury quickly, it undergoes a massive proliferative response, which renders the resource demanding. When you have a breach of the barrier, also renders the tissue higher risk of infection and genotoxicity. So our big picture question is, how do we balance opposing challenges and mount appropriate response? We propose there are 3 levels of response. So first, injury sensing induce a major repair response, which then is essential for repairs the injury site and lead us back to homeostasis. Secondly, injury also induce a quality control mechanism that remove the infected cells or cells that has been already damaged by genotoxic stress, while only allowing the healthy cells to repair. Thirdly, so local injury is able to trigger this systemic response that is essential for organism survival and fitness, and also feedback to the local injury repair, which is essential for homeostasis. So we will use combine biochemistry, mass genetics, or any technology necessary to answer this question at molecular and cellular levels. And with that, it's not only it will go beyond skin wound repair and also important for relevant pathologies such as inflammatory disease, infectious disease, and cancer. With that, I would like to thank my previous lab and also my current department, Department of Pharmacology and the CRI, and also Cancer Center. I want to thank my current lab members, and we are actively recruiting postdoc and grad students. I'm really thankful for my current collaborators that make this hypothesis happening. So thank you for my founding thoughts, and I would like to take questions. Thank you very much for listening.
Okay, questions. We have a question over here.
So very nice talk. So hypoxia is also known to induce the chemokines to promote the wound healing. I wonder if there are any crosstalk between IL-24 and the other chemokines during wound healing.
That's a good question. So we haven't really looked into it, but so IL-24 production is actually not only dependent on hypoxia and also, for example, STAT3 feedback loop. So I think it will be interesting to prove for the interaction between other chemokines and IL-24, but I'm not saying this is the only pathway that is essential for wound repair. Yeah.
Any other question? We have a question here.
Siqi, so does the state of cell division, so if you're actively cycling, does it matter? Cell division impact on the healing mechanism?
I mean, healing mechanism is a massive proliferation state, so it definitely matters how quickly you proliferate. Actually.
I wonder if you could comment on your connections between wound healing and cancer and how you think those might relate to each other.
So there is an old phrase back in 50 years ago that cancer is a non-healing wound. Injury state is, as I just mentioned, is one of the massive proliferative states in the adult, similar as cancer. So actually a lot of studies, including transcriptome analysis, they found that there are very similar response, both in cancer and wound healing at cellular and molecular levels. So there could be commonality versus difference between these two. So it will be interesting to figure out what their connections.
Okay, we have one more question.
Hello. Yeah. So I had just a small question for some cancer treatment, people inject intratumorally inject some drugs into the tumor. Will that produce some wound healing things? Do you think that is pro-tumor, anti-tumor in that sense?
So it depends. So basically what we propose that for a healthy healing wound, there is both pro-tumor, pro-proliferative and anti-proliferative mechanism that help only the healthy tissue to repair. So we suspect that in tumor, one of the surveillance mechanism is lost. Yeah, so it really depends, it's a context dependent. And I would say that it's interesting to explore both systems to understand that.
Thank you.
Okay, thank you, Siqi.